So the first type of microorganism that we're going to talk about is a prokaryote, right? So these are bacteria and archaea, and we're going to basically talk about them together because they have many similarities. So bacteria and archaea are prokaryotes. Prokaryotes, remember, they don't have nucleuses and they don't have membrane-bound organelles. So this is a prokaryote cell. And one thing that you'll see in a prokaryote cell that you won't see in a eukaryote cell are plasmids. So prokaryotes have their DNA in two places. They have chromosomal DNA, which is a large amount of DNA, and they'll just have it free in the cytoplasm. They won't have it in the nucleus, but they'll also have it in plasmids. And plasmids are little rings of DNA that can be used by the organism to make proteins, and they can be duplicated and shared with um, the the offspring of the bacteria, but sometimes not. They don't always get duplicated with chromosomes, and they can be shared. So bacteria can share these little plasmids like messages, and sometimes they contain really interesting information, like how to avoid being killed by antibiotics, right? So these guys can cause disease in humans, and they can tell each other how to avoid being killed by by antibiotics by sharing these plasmids. We'll talk about that a little later. Um, and they can move, and they also have pili. These pili will allow them to attach to other cells or objects, and that's how they end up sharing the plasmids. They attach to one another, and the pili can um, let them exchange this genetic information. Uh, Many prokaryotes also move with a tail-like extension called a flagellum, okay, which can rotate like a propeller. Most bacteria have a cell wall, and all of them have a cell membrane, and they all have ribosomes as well, and they all have cytosol. I didn't mean for it to be that big. There. Okay, so that's not much going on, right? Those are all of the features you can see in prokaryotic cells. It says in the below diagram, label the chromosomal DNA, the pili, the flagellum, the cell wall, the cell membrane, and the ribosomes. And then we're going to circle the plasmid. Okay, so take a moment and pause the video and see if you can fill them all in. Okay. So hopefully you took a moment and you paused the video and you filled them all in. So here are the answers. A are pili. Uh, Let me make that bigger. B are ribosomes. C is the chromosomal DNA. D is the flagellum. E is a cell wall. You can tell it's the cell wall and not the cell membrane because it's thicker and it's outside. F is the cell membrane. And here is the plasmid. So there you go. Okay. Let's talk a moment about archaea. So we said one thing that separates archaea from other organisms is that they live in harsh environments. And we have three different names of archaea, three different groups, and they are named after the type of environment that they enjoy. So one is called a halophile. Okay, halo comes from the same word as halogen. And halogen is a type of element that makes up salt. So halogens can uh, combine with metals and they can make salts. So halophiles, it means salt lovers. And they live in bodies of water where there is a high concentration of salt, such as the Dead Sea and the Great Salt Lakes. There are also um, thermophiles. Thermophiles are heat lovers. They live in very hot water, like in the ocean near volcanic vents. And we have methanogens. Methanogens are methane makers, and they live in environments with little oxygen. So they will do fermentation instead of aerobic respiration. And as a consequence, instead of giving off CO2, they give off methane. And these can be seen in landfills and also your intestines, right? Whenever you don't finish absorbing the nutrients from your food, these methanogens that live in your intestines, they finish it off for you and they create this methane gas, and then you get bloated. And that's why that happens. Now, we're going to talk a little bit about the reproduction of prokaryotes. Prokaryotes, they reproduce by a process called binary fission. Binary fission looks a lot like mitosis. And it actually has the same steps. The only thing that's different about binary fission is there's no nucleus. So you know the step in prophase where the nuclear membrane disappears. Well, these guys don't have nuclear membranes, so their nuclear membrane doesn't disappear because it was never there. So this is a type of asexual reproduction. Remember how mitosis, you got genetically identical daughter cells? Same thing with binary fission. They just make a perfect copy of their DNA. One goes to the left side of the cell, one goes to the right side of the cell, and then a cell membrane forms between them, right? So it splits into two identical cells. Because they reproduce asexually, there's no genetic variety. And this is a big problem for bacteria because then we can kill them all with the same means. Like we can introduce an antibiotic, and because they're all genetically identical, they all die. It's great. Um, but they have a strategy. So the strategy that they use to um, be genetically variable is called conjugation. And they can use conjugation to share genetic traits, okay, especially new ones, and alter their DNA. So, because binary fission works a lot like mitosis, the DNA from one parent is copied exactly to offspring. Unlike sexual reproduction through meiosis and zygote formation, this does not allow for genetic variability. Variability. So, how do prokaryotes alter their DNA? Conjugation is a process where cells with different genes line up beside one another and exchange genetic information. Conjugation can also involve the transfer of chromosomal DNA from one bacterial cell to another, and it can involve the transfer of plasmid from one bacterial cell to another. The transfer of a plasmid is shown in figure 6.7. This has a great medical importance. Oh, where did my diagram go? Well, it seems like it has disappeared. So let's look at a conjugation diagram. Okay, so here is conjugation. Right here. This is a good one. Okay, so we have two bacteria and they attach onto each other with their pili. And you can see there's a little plasmid. And once the pili have held on to each other, a little opening opens up, and the plasmid can go from one cell to another cell. So it sort of copies itself, and then the copy moves to the other cell. And then both cells have the plasmid, they have the extra set of instructions, right? So they can pass their genes on through this conjugation. This has great medical importance because some bacteria contain genetic information on plasmids that allows them to survive when exposed to drugs that should kill them, like antibiotics. So bacteria that are resistant to drugs pose a health risk because the diseases they cause cannot be easily cured. So here we have two diagrams, okay? And one shows binary fission and one shows conjugation. So you take a look at both of these diagrams, okay? And you decide which one is binary fission, which one is conjugation. So the top one is binary fission. Binary fission, because you can see the two are lined up and one is passing genetic information to the other one. And you can see the bottom one is wait, did I say binary fission? Yes, I did. I meant conjugation. So one is conjugation and the bottom one is binary fission. So conjugation shows the passing of uh, chromosomes from one to another, and binary fission shows the cells separating into two cells. And that's really it on bacteria. So for homework, you are going to read a few little short passages about bacteria, and you're going to answer some multiple choice questions. You can see the answers at the bottom of the multiple choice page, so you can check your understanding. Okay, and then we have a second worksheet that will tell you a little bit more about bacteria, right? Some of it will be the same as what we've covered. Some of it will be a little bit different. Okay. And then we have some questions that you can answer to practice your new knowledge. And then this right here on page 11 is really important, the classification of bacteria by shape. So you're going to definitely read this because you're going to use that on your assignment. Okay. And then, so there are some more questions at the end of this reading. And then you have another reading. Okay. And then you have an assignment called Shapes of Bacteria. So you are going to get your answers to this from page 11. Okay. You're going to use page 11 of your notes package to find out the meaning of each term. And then you're going to combine the terms in order to describe 1, 2, 3, 4, 5, 6, 7, 8, 9 different types of bacteria. So some of them will just have one name, and some of them will have two names combined together. Okay. If you read page 11 very carefully, you will know how to do this very easily. And that's it on bacteria.